faithfully conveying value, interpreting global great books. Hi, this is Sally's Book Club. Today we'll be exploring the book Missing Microbes, subtitle The Health Crisis Caused by Antibiotic Abuse. What if I told you your body isn't entirely your own? Would that surprise you? In reality, each of us is a superorganism containing not just 30 trillion human cells, but over 100 trillion microbial ones. These invisible microbes mostly reside on our skin and gut, working symbiotically to digest nutrients and fight diseases. They also help train our immune systems and reduce allergy risks. So, behind every healthy individual, there are microbes working tirelessly as silent guardians. Yet, over recent decades, these microbes have faced unprecedented challenges. C-sections deprive babies of their first microbial inoculation from the birth canal. Meanwhile, antibiotic overuse disrupts microbial development. This microbial depletion links to modern ailments like asthma and IBD. How did this crisis emerge? And what can we do about it? Missing Microbes provides the answers. Author Martin Blazer is an NYU medical professor and National Academy of Medicine member, specializing in microbiology. In 2015, Time magazine named him among the world's 100 most influential people. So, this book unravels the complex relationships between bacteria, disease, and health. After listening, you'll reconsider C-sections, antibiotic use for colds, and food safety issues, empowering better health decisions for your family. Next, I'll explain the human microbe connection through 4 sections. First, understanding our bacterial ecosystem. Second, the history of antibiotics. Third, health crises from antibiotic overuse. Finally, solutions to mitigate this crisis. The book's title is Missing Microbes. First, let's clarify. Microbes refer to microscopic organisms invisible to the naked eye, including bacteria, viruses, fungi, and more. Some people confuse these 3 types of microbes, but distinguishing them is simple. Both bacteria and fungi are self-sufficient, complete organisms. They differ in cellular structure, while viruses are genetic material carriers that must hijack living cells to reproduce, like influenza viruses causing flu. This book focuses on the disappearance of bacteria. First, let's examine our bacterial ecosystem. Modern society's obsession with sterility has become deeply ingrained. While many view bacteria as enemies, some are actually our allies. Our bodies host bacteria in the gastrointestinal tract, skin, nasal passages, densely populated microbial communities. They're so tiny that 1,000,000 bacteria stacked together would be the size of a pinprick. How abundant are our bacteria? Skin bacteria could form a pea-sized ball. Digestive tract bacteria would fill a soda can. Thus, each human is essentially a superorganism carrying massive bacterial colonies. This might sound slightly gross, but don't panic. Bacteria aren't scary and can be beneficial. Bacteria colonized Earth 3 billion years before humans appeared. Through coevolution, many bacteria have become our ancient companions. Our first bacteria come from the mother's birth canal. Lactobacillus and Bifidobacterium colonize infants' guts, creating environments for beneficial microbes. As children grow, diet and environment shape mature microbiota, forming diverse ecosystems like rainforests. Throughout life, we provide bacteria warm habitats. They reciprocate by safeguarding our health. Human bacteria serve 2 main functions. First, aiding digestion and nutrient processing. We often hear eat more whole grains and vegetables. Actually, the dietary fiber in these foods can't be directly digested. Our gut microbes process it into absorbable nutrients. Moreover, gut bacteria produce short-chain fatty acids and vitamins. They're essentially our body's nutrient factories. Secondly, bacteria protect us from diseases. Beneficial bacteria crowd out harmful invaders, acting as our first line of defense. They also train our immune system to distinguish friend from foe. Bacteria are immunity's first teachers. Newborns arrive with no immune system. but already hosting their first bacterial colonies. These pioneer microbes from the birth canal and breast milk create environments for more beneficial bacteria. As the immune system develops, it recognizes these bacteria as part of the body. This microbial exposure builds immune system expertise. When microbial balance breaks down, the immune system attacks harmless substances. This can range from seasonal allergies to severe conditions like asthma and rheumatoid arthritis. In part one, we explored our microbial ecosystem. Bacteria are ubiquitous guardians of health, but among these microbes exist harmful pathogens too. These dangerous bacteria once threatened human survival. Now let's explore humanity's battle against pathogenic bacteria. Historical plagues nearly caused human extinction multiple times. The 1347 Black Death wiped out one-third of Europe's population in a decade. The 1918 Spanish flu infected 500 million globally, killing tens of millions. Before 20th century, diseases like tuberculosis and typhoid defeated rich and poor alike. For instance, in 1884, typhoid fever ruthlessly claimed the life of 15-year-old Leland Stanford Jr. His father was the governor of California. The grieving governor and his wife established the world-renowned Stanford University in his memory. After millennia battling infectious diseases, science finally made a critical breakthrough. In 1857, French scientist Louis Pasteur proved through broth experiments that invisible microbes cause decay. Building on this, 19th century scientists developed germ theory, linking diseases to pathogenic microbes. Using staining techniques, scientists could observe and culture bacteria under microscopes. Early attempts to kill bacteria included dyes, poisons, and heavy metals, mostly ineffective. Some doctors even used bloodletting, which often accelerated patients' deaths. Penicillin changed everything. Humanity began turning the tide against infectious diseases. Discovered by Scottish scientist Alexander Fleming during his bacterial research in 1928, returning from vacation, Fleming noticed a petri dish contaminated with penicillium mold. Remarkably, the mold killed surrounding pathogens, revealing its antibiotic properties. Fleming isolated the active compound, naming it penicillin, but initial yields were too low for practical medical use. The World War II's outbreak in 1939 created desperate need for infection control. Oxford scientists began industrializing penicillin production in 1940. The research relocated to America due to Britain's wartime conditions. Penicillin's 1942 clinical debut showed miraculous results. A 33-year-old American woman with streptococcal sepsis recovered after near-fatal post-abortion infection. After receiving 5.5 grams of penicillin, the woman regained consciousness within hours and fully recovered in under a month. This miracle cure-like efficacy ignited medical enthusiasm. In 1943, scientists isolated a high-yield penicillium strain from a moldy cantaloupe. Kaiser's mass cultivation methods ushered in the golden age of antimicrobial development. These bacteria-fighting drugs became known as antibiotics. Fundamentally, antibiotics are nature's biological defense weapons. Through evolution, organisms developed unique metabolites to combat competitors. Antibiotics, primarily produced by bacteria and fungi, exemplify this evolutionary arms race. Some disrupt bacterial cell walls or protein synthesis. Others inhibit growth directly. Penicillin works by breaking bacterial cell walls, causing them to burst. Modern science has discovered numerous antibiotic varieties. Chemical synthesis now supplements natural antibiotic production methods. Antibiotics revolutionized treatment of meningitis, tuberculosis, STDs, and more. They became universal solutions for bacterial infections. Modern surgery would be impossible without antibiotic protection. Surgical infections could lead to amputations or death. Cancer treatments like chemotherapy depend on antibiotics to prevent lethal infections. The author nearly died from Salmonella typhi infection before antibiotics saved him.